This interview with Dr. Allison was recorded at the 2023 NFCR Summit and repurposed for this podcast.
This is All Things Cancer, the podcast of the National Foundation for Cancer Research, hosted by NFCR's President and CEO, Dr. Sujuan Ba. Dr. Ba is a globally recognized cancer research advocate and visionary leader. All Things Cancer episodes will explore the hindsight, insights, and foresight of cancer, from research discoveries to patient care advances, and how NFCR and our collaborators are working to accelerate the process of making cures possible.
Hi, I'm Dr. Sujuan Ba, and in this episode, I'm joined by one of the most influential scientists of our time, Dr. James Allison. In 2018, Dr. Allison won the Nobel Prize in Medicine for his work in immunotherapy, and before that, in 2014, he won the Albert St. George Prize for progress in cancer research from NFCR. His work uncovering how to activate the body's own immune system to fight cancer has transformed treatment and given hope to countless patients worldwide. In our conversation, Dr. Allison shares what first sparked his scientific curiosity, the challenges he faced in pursuing ideas that many thought would never work, and the personal experiences that fueled his determination. This episode offers a personal and inspiring look at a scientist who never gave up and whose work is still changing the future of medicine. Hi, Jim.
Hi.
So honored to have you here with us at the Global Summit and award ceremony. It's been a while since last time we see each other.
Yeah, it's been a while. But
Yeah,
I'm glad to have a chance to come back to this 50th anniversary meeting as well.
It's fantastic. And we were so proud when you were selected as a St. George Prize winner back in 2014. And before you got all other major awards, including the Nobel Prize winner 4 years later. So could you share with us what the St. George Prize means to you?
Well, the thing that I think that St. George Prize meant most to me was that sort of in part was validation of the science, because you probably know immunotherapy was not held in the highest regards by many people doing cancer research for a long time, particularly those that were involved in precision medicine. The St. George Prize had a history of going largely to people who made contributions in those other areas and not in immunotherapy. To me, was the first time, was one of the first times that there was really validation saying, oh, this works in cancer. That was in addition to personal satisfaction, of the honor of such an organization. It wasn't just for me. I think it was for validation for the whole field.
That was the first time the whole Blue Ribbon Committee members unanimously believe you have made such an incredible breakthrough contributions to immunotherapy and to cancer patients. So we were so thrilled about that. And actually, as time goes, many people think the St. George Prize has become a bellwether for other under-recognized people and recognizing the pioneering researchers.
Yeah, that's certainly
For the mainstream recognition.
Yep, exactly.
And you mentioned that early years, you really struggled to get funding for your immunotherapy research. And I'd like to hear you share some of the reflections, like how do you come up with these thoughts of exploring the body's immune system to fight cancer? And how does that evolve from the beginning to today's the approach? Almost every other drugs are trying to be in combination with the checkpoint inhibitor.
Well, I trained in biochemistry, so I, like everyone else, was thinking of chemical approaches and this sort of thing. But good fortune to hear a lecture when I was an undergraduate, actually, on immunology, and T cells had just been discovered. I'd never heard of them. I heard antibodies, of course, everybody knew about this, but T cells were a new thing, and I could understand antibodies, these things that are made like enzymes, because I studied enzymes, have a specific site they can go and, bind to things. And
That's such an interesting turning point. Hearing that lecture and being introduced to T cells for the first time. So, what was it about that moment that really captured your curiosity and made you want to learn more, Jim?
T cells were a whole different matter because they're cells, and they go through your body, go in the blood and the lymph, but they also percolate through your tissues, looking for viruses and other things that ought not to be there. And that's all they knew when I first heard this lecture. That was essentially the entirety of the lecture was being told that that's what they did. And when I asked the professor after class, well, how do they do that? How do they recognize? What are the signals? What are this? So we don't have a clue. In fact, he didn't even think there was such a thing. He thought it was just, another kind of blood cell that happened to have an antibody stuck on its surface. It was a passenger being carried along. But I decided that I wanted to understand them just because of the puzzle of it. How do you have a cell that can go around your body looking for things that ought not to be there? First of all, this concept of not self, how do you recognize not self? And so that was the mystery. What sort of molecular, it has to be a molecule. That was it. But then how do all these other things that they do, how does all that work? I thought about that first, and then, of course, it occurred to me that if we could ever understand that properly, maybe we could understand how to control it sufficiently to get them to attack cancer cells.
That's really a profound scientific curiosity, wanting to solve the puzzle of how T cells distinguish self from not self. But was there also a personal element that shaped your motivation?
I've had a lot of cancer in my family. My mother died when I was 10 from lymphoma, and I saw what radiation does to you. Her brother died of lung cancer, and I saw what chemotherapy did to him back in the early days when, there weren't many successes. And, I just thought, well, that wasn't, I guess what I thought first was I'd like to figure it out, and then if I could, figure out some way to do it. Every now and then I would make enough of an advance to try a few things. And I didn't really have any trouble getting funding for the basic science. But when I started saying, well, let's try this to treat cancer, that's when the problems arose, because everybody said that'll never work. People have tried that, and it's never worked. So, just stick to the basic science. That was the hard part of it because, I got funding for the basic science. The idea was sufficiently controversial that drug companies wouldn't touch it. The pharma companies wouldn't touch it. They said, immunotherapy doesn't work, period.
And the government?
Well, the NCI was a little bit different, is a little bit different now. That was a question I had. I went to the director of the NCI at the time, asked him and explained my dilemma. I said, I've got the grants, and I know that it's a work, but I can't do anything with it because it's not fashionable to commercial operations. Why doesn't the NCI have something to help people make the drug? First they said, oh, well, we can't do that. We're to do science, not the drug. We're not a drug company. And I said, anyway, that actually led in part to development of what was called RAID, the Rapid Access to Interventional Development program that the NCI started in response to. By the time they got that done, I'd finally, after 4 years actually of trying, convinced the company to help me make the antibody, just to make. I made the thing to do all the animal studies, but making one for clinical grade, of course, is a whole different story.
Yes.
I simply did not have the money.
Very
I couldn't get a grant. It's very expensive, but I couldn't get a grant to do that.
Yeah, that's a proof of concept.
Right, and
The
so and the
preclinical
company,
studies.
and so I went to every major pharma, and I don't know, I can't remember how many biotech companies, and a lot of times they just say, "Oh, it's never worked, and it never will." And if I heard that from a scientist, from a businessman or a lawyer, I think, well, that's okay. You're just ignorant, But when I heard that from a scientist, I found it insulting. I said, "You're a scientist, and you're going to sit here and tell me it'll never work because it never has." Well, tell me an experiment I can do. Tell me, because that's not, as a scientist, I don't accept that level of just blind skepticism or rejection of something just because it's never worked. I mean, that's not a reason. And anyway, so that was frustrating and maybe bad. Sometimes some other companies, it got up to the level of them thinking about it, but They just thought that, it really wasn't something that could sell well, or they were worried about patent situations and all this. But finally, it just so happened there was a small biotech that didn't have anything else to do, to tell you the truth. But they had a mouse where they'd replaced all the mouse antibody genes with human, but they didn't have an application. I said, we were introduced to a friend. I said, here's the application. I need a human antibody. And so they used their mouse to make the human antibody. And actually, the first money came from the Prostate Cancer Foundation in part. And finally, that company got to phase 1, and in the first trial, there were trials all over the place, and there were a few successes, but the one that got everybody's attention was a phase 1 trial in stage 3 and 4 melanoma, which was lethal. And no drug had ever extended life against that. There was no drug at all for treating it. And they had 3 out of 14 of patients in the phase 1, which is just a safety trial, that all their tumors disappeared. And so then that sort of broke the back on everything, because nobody could say this isn't going to work after that.
That is truly a breakthrough moment. And you mentioned a couple of points. One is, in order to do breakthroughs, have high impacts in science, you really have to ask questions, have consistent curiosities. So your curiosity about the T cell, the body immune system, started with you when you were an undergraduate student. That's amazing. It stayed with you. That is incredibly important message.
Yeah, that's about 1968. I thought, well, I'm going to figure out T cells, and here it is 50 years plus later, and I'm still trying to figure out how T cells work.
And that was usually the case. The curiosity of the wall, curiosity usually leads to incredible breakthroughs like what you have seen. And another message you mentioned, government has a lot of basic science funding. The commercial, pharmaceutical companies, VCs have a lot of investment for the technology, the molecules already in the late stage. It is this early stage, right, from the discovery you made and to the time you convince somebody take like your
Some people call it the Valley of Death.
Valley of the Death, exactly. And that started when you were doing that 1980s?
1993.
1993. And took how long for you?
It was until 98 to get a company that would make
For
it.
5 years. Took 5 years to convince a company to recognize the value of your discovery and to be willing to take a risk to carry through the preclinical, the Valley of the Death. That's incredibly unsatisfying, frustrating, because the patients can't wait for 5 years. And the 5 years waiting time for somebody to do something with your discovery, how many people have died? And that is actually one of the problems NHR has been trying to address. And that's how we, NHR give the capacity building grant, and we set up the Aim High Accelerator Fund. And we wanted to provide those funding for the scientists like you who made the discoveries, and then they needed to show the proof of concept,
Right.
Get through the preclinical trial and get to phase 1. And that is the hardest.
And you need to have a situation where it's scientists making those decisions, not lawyers and businessmen. I think it's something. I'm not trying to criticize them too much. I mean, you have to have them to really develop it finally, but there needs to be help for taking the ideas that people have and seem good, but don't seem obvious necessarily to somebody who needs to make money to the lights on, and that can help get to the proof of concept, as you said. So
It's the hardest thing.
and that's really where we need more help. And there's a lot of, well, I should say a lot, but there are many disease-specific organizations that'll do that. They'll do it for prostate, they'll do it. Even something just as broad as all cancers is really not, there are a few, but not very many.
True. That's actually is a challenge. We as an organization that is focused on all cancer, not just a specific disease-related cancer organization. That was one of the challenges we had in terms of getting the public support, getting resources, because people identify their disease types, affinity. If they have their daughter or wife have breast cancer, they are going to support breast cancer. But we have always been telling the public cancer is a genetic disease. So when you actually demonstrate the immunotherapy theory that is about the whole body, and that actually validated some of our early beliefs.
Yeah, for a long time, the idea that, we need to get the genomic sequence and then to try to understand, because obviously cancer is a genomic disease. It's not, somatic cells, not germ cells, but it's obviously a genetic disease and caused by mutations. So the focus for a long time was that there's three types of mutations: the drivers, the actual mutations in the protein that says be a cancer cell, start dividing, then the elimination of the suppressor genes, for example, the kind of cells that say, well, if you get too weird, you're going to die. You lose those. You gain a function there, you lose a function there, and you get cancer. And then the third bin is everything else. And so I remember people used to say, well, that's just garbage, or those are passengers and they don't matter. But what we know now, and this is something actually that I was thinking about really pretty early in the process when I saw some data from Bert Vogelstein about bowel cancer, and that there were very few drivers, but a lot of passengers, because the immune system doesn't care. And the immune system detects mutations as well,
Yeah.
mutations that change an amino acid, but it doesn't matter to the immune system whether it turns protein, which normally, has an on-off function, and when it's locked on, I mean, the immune system doesn't care. It just knows it's not self.
Yeah.
It knows it's foreign, it shouldn't be there. Those are what it's not that the immune system focuses on those, it doesn't care, it's agnostic. It sees all the mutations.
That's such an important distinction. How the immune system reacts to mutations differently than how we traditionally think about targeted therapies. So can you explain a bit more about how that understanding shifted the way we think about treating cancer, and particularly with immunotherapy?
In a way, the old precision targeted, genomics targeted therapies are like, one of the things in melanoma, for example, that made it so hard to treat was that they're so genetically unstable. You get 60% of everybody's melanoma is caused by a mutation in a molecule called BRAF that tells the cell to divide. And so, you can make the perfect BRAF inhibitor and you can kill 99.9% of the tumor cells. It won't matter because there'll be an activating mutation in MEK, which is downstream of it, or another mutation in another pathway, or this or that. There's so many mutations that's why those conventional, more what I call, they weren't conventional at the time, but the usual way of doing precision medicine, you identify the target, go after it. Is not curative because there's 17 different ways I've been told that a tumor cell can get around B targeted BRAF.
Yeah.
One drug. And so you're going to have 17 different drugs, But if you can get the immune system to see a melanoma cell, the average melanoma cell has depending on the kind of melanoma, may have 300 to 2,000 different mutations per cell. And many, not all, but many of those will be probably somewhere around, 20% or so might be targets for the immune system. And so you got so many targets for the immune system. And
that's the challenge, of dealing with cancer. It's really, truly genetic mutations constantly.
But what that meant was, though, that the disease is like, it had a lot of mutations like melanoma and lung cancer that were so hard to treat by the more what's called precision medicine. They're sitting ducks for the immune system because they've got so many mutations. They've got more shots on goal, if you want to put it that way, for your T cells to attack. So, it's kind of a dichotomy, But also it makes a case for a way to make, for example, a drug that has a single specificity into one that has a lot. Because if you could go in and kill, I don't know what the number is, let's say 50% of the cancer cells, that's enough to prime an immune response that can take out the rest of them by reacting with the other mutations that weren't in that first one. It had nothing to do to distribute it.
Right now, there's so many treatment approaches. Use the combination of the, target therapy or chemo in combination with immunotherapy. The drug you discover and you champion all the way to the patient's bedside. How do you feel about that? Seeing that finally gets to
people
side.
Well, people used to say to me, "Oh, well, we could do away with chemotherapy. We could do away with this stuff. We could do it all with immunotherapy." But I don't think that's true completely because there's nothing that starts an immune response, a T cell response, better than having an inflammatory death of some cells. And so you can accomplish that with chemo, and you can accomplish that with some of the, at least of the genomic agents you could do with radiation. But so I think that the cancer treatment of the future is not gonna be any single one of those. It's gonna be the combination.
Yes.
And, if you've got a good drug that kills a lot of lung cancer cells, but not all, and isn't curative, as long as it doesn't inhibit with anything the T cells need as they're developing, you can put it on top of it and have a more than additive function. I mean, we're beginning to see that. With melanoma, for example, right now, by strictly using two immunological drugs, one to CTLA-4 and one to PD-1, the survival rate is somewhere above 55%, where prior to 2011, prior to the advent of immunotherapy, the 5-year survival was less than 3%.
Yeah,
And
that's
there was no drug that ever changed that. But now we're up to 50. Let's just say 50. But there's some new trials combining chemotherapy, surgery, and the immunotherapy given at the proper time, where in one trial, and it was relatively new. It's only 3 years old, but the 3-year survival was 77%.
So that's
All
an
those were
incredible
fully responsive.
improvement.
Yeah, and so
We still have to get to up to 90%.
good if we can, why not?
Only by then we can say it's a cure, right?
Yeah, but that's where, and that through the work of the foundation and others, we've gotten to this point. But the optimistic thing about it is that some cancers we can cure, or at least extend for years now, the survival. I don't know when you call it cure. People say the formal definition of cure is that you don't have a single cancer cell in your body. Well, that's not that's not a useful criteria. But let's say 5 years, 10 years without therapy.
Survival. Yeah, survival.
So by that, we're curing about maybe 20% of all cancers.
That's incredible progress.
Some of them are none, but it's on the high end, like I said, in melanomas up there. Lung cancer is what, 30 to 40%, bladder cancer is 30% or so. And melanoma is caused by ultraviolet radiation of the sun. Those others are all associated with smoking. Those all have a lot of mutations, a lot of targets for the immune system. It's going to get harder as we get down to smaller numbers. That's why we're going to have to start relying on, to get the immune system kicked in, we're going to have better ways of, combining things. Anyway, but we're nowhere on glioblastoma, for example, and pancreatic.
And that's one thing we at NSR is very dedicated supporting that almost like one of the few last territories that we have now overcome. And that's why we are part of the supporter, organizer, you call it, for the GBM Agile.
Yeah.
Yeah.
That's an important one. But the good news, is we know it can be done. And the question is just how do we do it better? And so we need more research. Now we know how to focus on some areas that.
And we also need more organizations to take risk, because a lot of discoveries to fund that went from discovery to the preclinical. Yeah. So Jim, you mentioned when you first discovered the molecules, you believe that's going to be very important for treating melanoma and really activating body's immune system. But it took you 5 years to convince a company to take on that risk. And that was 1998. And still now, so many decades later,
it's still the same situation. What do you think you have the advice for investor, for the scientist, for the greater community?
The scientists or the guys on the other end? First of all, I wouldn't necessarily say go try to cure cancer. I'd say do what you love to do, and but pay attention to what you're doing. I mean, the way I went about it is I didn't get up every day saying, "Oh, how can I cure cancer today?" I didn't do that. I said that actually. I was interviewed soon after I went to Memorial Sloan Kettering in New York, and I said that, and they actually put it on a photograph of me on a placard in the institute, and then everybody I ran into said, "Well, what the hell do you do then? I mean, you're at a cancer center, you know?" I said, "What I try to do every day is understand how the immune system works in detail so that I can think about how to use it to attack cancer, from a knowledgeable, informed way, it's a
That's the byproduct.
lifetime, yeah, I mean, I could tell you how, this will go in and it can make this happen to
You love
make your
science.
cancer.
You love science
Yeah, yeah.
exploration.
But, I mean, so I think scientists, I would just say do I mean, I would get inspired by trying to figure something out, and then every now and then I just quit thinking about what I was gonna do next and say, "How can I do what are the experiments I need to do now to take that observation and try to move it into the cancer," because you have to shift gears to do that. So how do I shift thinking that way before I go back and dive into the bore into the part of how T cells work again?
So the tenacity is key. Tenacity
Yeah.
is key. Yeah, don't give up.
Don't give up and, pay attention to what you're trying to do.
I love it.
Yeah,
I
and love
dig
it.
it. Yeah.
On the other side, I would say, we're a little bit we've gotten past that now, so everybody, understands the power of manipulating the immune system. But what I'm afraid is that it's all shut down now. People say, "Well, this is the way to do it." And there's a lot we don't know about how to do it right now, which is why a lot of what's being done now is gonna fail because now they're beginning to skip a step, like, because they don't wanna take it again. Before they'd say, "Well, that's never worked." And now this works, but if you've got something really new and it's never been done, then people tend to say again, "Well, that'll never work." And we're back at, the paradigm has just moved now to where it includes immunotherapy. But, there's a lot of trials going on. For example, say, you have this immune checkpoint and that immune checkpoint. You put them together, now to work. Except what they don't know is that sometimes that second one is one that's not induced until, you give another one, and the whole tumor situation changes depending on what the T cells are doing. The T cells change too. The environment changes, then the immune system changes with it. So we put in that first antibody and change it, then it's different. And if you think, well, here's another immune checkpoint that works in mouse models, really, but that one may not even appear until you've given that first one, and yet there are people that try to test it as a single agent and see if it's gonna work. It's got not a chance of working because it only appears. People look at it and it's implicated something downstream, but without knowing the timing of these things and what it takes to go up and to come up and the things that are present when you start perturbing the system.
And so your point is be innovative. Always.
Well, just like now, I would say just we need to have a trial where you just spend, 3 years looking for a clinical endpoint. If you don't learn something about mechanism, is a waste of time because chances are you're gonna fail.
Mhm. Yeah.
Even when you're well informed, you're gonna fail. 90% of the time you fail. So if you don't wanna fail, you need to do the science first and see what's going on.
My thinking is all those failed trials, failed experiments can infer some good learnings. And that
They
could
they
guide
could they
us
could
for
if they
future design of the new science and scientific projects and also new trials.
Well, if you look and see why it failed, because if you conclude that that second target that that target that you tried, it didn't work because it's not expressed necessarily in the right time with the rest of the immune changes that are going on, and all you do is look for a clinical signal, you're not gonna learn anything except it didn't work, and then you might throw that drug away when it was really perfectly fine in another context.
That's actually is important philosophy you are talking about, and many organizations have very short term, philosophy. Every several years, didn't get the result, that flip it and go on the new things. At NFCR, we provide long term, sustainable, flexible funding to scientists. That we don't just look at, okay, 2 years didn't work, move on, and we believe in the scientists. If they are doing the good science, we follow them.
That's very important, because one of the things that everybody in this field knows is that of course, the most important source of funding for all this basic science, for all this work, is the federal government, the NIH and the National Cancer Institute. But their grants, until you become very senior, are good enough to get one of the merit awards, where you get a 5 or 7 year funding period. Is 3 years, which means you got 2 years to put in a renewal. And there are a lot of things that you can't do from start to finish in that amount of time. And so it does take, I think, like you said, the funding sources that will have faith in the investigator and the quality of the work, without saying you got to have a killer experiment every 2 years.
Correct. Yep.
Not too many people can do that.
Right. That's actually one of the traditions NFCR has kept since the day of St. George, and that was one of the recognition that we have.
That's very important. That's critical. Yeah, not and too many, even the other sources of funding cancer, the foundations are looking for instant returns.
Yeah, but science is not instant.
I mean, that has its place. If somebody has something, like your work gets to the point where I was in 1995, we could have gotten an answer really quickly. All you need to do is make the drug. It was obvious. Do this, do that, and you'll get an answer. And most of the time, you're not there, though. You think, oh, well, it might, but I'm not quite ready to bet on it completely, but you're going in the right direction. And so at some point, you need to give somebody $1 million to do that, the one-time thing. You're going to either work or not. You're going to get an answer up or down very quickly. But the other stuff, you just got to have patience and keep funding them, and they keep working on it. Or maybe they change, They say, okay, that idea was bad, but here's my other one. And it's,
But,
that's happened to me several times.
Yeah,
I'd spend 2 years on something, or 3 years, and then I'd figure out, no, and just dump it and go work on something, move over. Well, then I quit on immunology, just quit on that aspect and move to another issue, And
But a lot of government funding doesn't allow you to change the direction of the funding in the middle of the funding period, right?
yeah, all you can do is not ask for renewal and write another grant on your new stuff. And you could keep doing that forever, but you're not going to make any progress.
Exactly. That's exactly the point. And that is another thing NFCR is different, is since we work with the scientists so closely, and when they have a change of direction, when they have unanticipated discoveries, they come back to us. Even when they are in the middle of the funding period, and they come back and they ask whether we could redefine the direction, and the other didn't work and we have new direction, and we allow that flexibility. More people believe in the tenacity aspect of funding to support the blue sky science, and more chances we get breakthroughs.
And to get more people trained as to how to do it that way.
Right. And ask, so encourage people to ask a big question, not to worry about my funding won't be renewed. Yeah. So
If I can't settle this in 2 years, then, I'm not going to get a renewal. And so then you just prove something, even if it's not very important.
and that is such important message. I think not only the government policy people need to know, the public, general public need to hear.
Yeah, well, I think that funding, people that have a demonstrated record of good ideas and stuff, even if some of them haven't been home runs yet, if they're making steady progress, and you have confidence that they're on the right track, they're trying hard, and they've got making progress towards something that's measurable and have a clear sight of where they want to go. I think you've got to stay with them for a while.
Yep.
I could argue that the first experiments that I did along this, I did in 1981.
Wow.
Yeah.
And I also wanted to get your thought about the future. What you are most excited about the current landscape, and what you are most excited about the future of cancer research.
I'm excited now about what we're seeing in combinations with the fact that uh, by combining the different modalities, I mean, we've known if you can cut tumors out, we've known that since the Greeks, 6,000 years probably. With radiation, we've known since Madame Curie. Of course, surgery, if you could cut it all out, you win, but most of the time you can't. Sugar metastatic disease and then radiation, cured a lot of people, but the side effects of that. And then chemo came about after World War II, with chemical warfare agents and chemical warfare things, and they can poison cells, and they poison fast dividing cells and all this, and they cure some cancers, but drawbacks to all of that with side effects. And then finally, there's the genomically targeted things we've got now that have the problem of they can kill some cells, but they're not going to ever get all of them. We know now that you could combine, what I'm excited about is you can combine surgery, even if you don't get all the tumor. There's some studies now showing very clearly that patients that get one big metastasis removed and have immunotherapy before and after do better than the patients who had either alone, that had just the immunotherapy or just the surgery.
That's
It's too early to say if there's any
right.
cures there. But there are some clear studies where you combine chemotherapy with surgery and immunotherapy where, there's really an incredible increase.
It's remarkable to see how the science has evolved, not just in understanding individual therapies, but in realizing the powerful potential of combining surgery, chemotherapy, radiation, and immunotherapy to achieve better outcomes for patients.
And the cool thing about those is it looks like you can drop the dose because you don't have to keep doing it. With previously, the thing that was bad about chemotherapy and radiation was you have to give it till you kill that last tumor cell, and you don't have to do that anymore. All you gotta do is kill enough to prime an immune response and let the immune system take over. And so we can drop the bad side effects of those if we just pay attention and more. Then, of course, there's at least a dozen targets, specific targets just for checkpoint blockade that I'm aware of that may be important too, but we need to. But it's complicated, and they're on different kinds of cells, and they come up at different contexts at different times of the response. So we need to really study the biology of those before we can start making appropriate combinations, and then adding them to the mix. To me, the most exciting thing here is it's, let's pull it all together. Let's get an integrated view of cancer therapy. Let's not talk about radiation therapy or chemotherapy or targeted therapy or immunotherapy. Let's talk about cancer therapy. Do whatever it takes to
I love that.
kill the damn stuff.
Yeah.
I mean, that's what we want to do.
Cancer therapy.
It's cancer therapy,
Yeah.
and whatever it takes. These individual. It was useful in the past. It's useful
That you
to know their
learn.
differences because
Yeah.
once you understand how different they are, then you know how to put them together properly. So
And
that's sort of
that is really uniting all the aspect of the approaches, right, to treat the cancer. That's wonderful. Thank you. I know you have such a productive long career and made a lot seminal discoveries. And can you share with us the moment that you feel most proud and about what you have done?
Well, there were two moments. One was when I didn't see it, but when a graduate student first brought me there was an experiment that even my own students thought had such a small chance of working. They didn't want to do it. Finally, I talked a guy into doing it and came back, and I was expecting we'd cure a few cancers, but we had a big experiment. All the mice were cured 100%. This experiment was beautiful because they were all dead or they were all alive. I mean, there was no dichotomy, and then we did that again. That was important. But I think the best one was when I got a call from my colleague Jed Walchuk when I was at Sloan Kettering.
Sloan Kettering. Yeah.
And he said, "Come down here, Jim, to outpatient." I said, "I'm busy. It's like 5 blocks." I mean, it wasn't that busy, but it was summer, and I didn't want to walk 5 blocks, But anyway, so I decided to go and went down, and I walked in this room, and it was him and this woman. And I walked in the room, and she just grabbed me. Anyway, it turned out she When she was 22, she was diagnosed with metastatic melanoma. She had 31 mets in her lungs. She had a brain met, and she had God knows how many in her liver. And she failed chemo, 2 or 3 different chemo. She failed a couple of targeted therapies. She failed radiation to her brain. And she came in, one of the first people to get the drug that I developed. And I moved from Berkeley to Sloan Kettering, away from the basic science to the clinical, so I could be there, and hopefully contribute something. But anyway, but her tumors had vanished, and Jed told me that, keeping her barely alive, and this time she'd failed everything, and they said, she was hospice bound, and the last thing we got is this new drug, Trithid. So anyway, she took it, and a few weeks later, she went out, she came back and had the scans done, and Jed said when he'd called the radiologist, he said, there's something wrong because there's no cancer in these films. There must be, did you get the right patient? Anyway, they had, but she was completely, and that was in 2006.
Wow.
And now she has 2 kids.
Isn't it wonderful? That is most gratifying moment. She's still, well alive. But because of the drug,
they made all the mouse experiments. I was thinking when I went to lab, I said, I pick them up, they try to bite me, They don't know I just cured them of cancer, they try to bite me and pee on me, and, of course I gave them the cancer in the first place, so it was a fair trade, I guess, but anyway.
It's wonderful.
But that was when it finally dawned on me, this really worked. So Sharon and I became good friends.
The science we support not only is good, important for cancer and for other diseases, and then, for other fields.
The thing now, we know we could cure cancer. We didn't know that 15 years ago. We know now we can cure some cancers. And so the basic rules. Now, maybe we're going to hit a point where, well, right now, with pancreatic and glioblastoma, but I think there's some hints of things to do there, but I think now is even more a time to do good science. And analyze, not to go way beyond just doing. There aren't enough people to do all the combinations that are possible now. There are not enough patients to do those trials. We got to do the science up front and de-risk these trials with mechanistic data before we can go. So that's what we need to do now, is fund more of that kind of research where we keep trying to discover new targets for, genetic targets, immunological targets, whatever, but develop stuff, vaccines, whatever it takes, but also pay attention to that in between that we talked about.
Yeah.
It shouldn't be the valor of death, but it should be the value of ignorance either. that needs to be filled in with data so that you do small trials in patients and analyze the tissue. And did we get there or not? Because now you can't just go from animal experiments to do 1,000 patients and something and look at statistics and ask the question, did some people survive more than the median? That's so just retrograde to me. We shouldn't be doing that. You have to do that at the end to prove it works, but that should be the final test. And you ought to know the damn stuff has a 90% chance of working before you do that. And the only way you're going to do that is to look for the changes that we know what to look for now, to a large extent, not completely. We need more work on that too. But with the advent of single-cell RNA, single-cell proteomics, spatial now, we can even take a slice of tumor and tell you what every gene makes and every cell in there and point at them and say, is this T cell next to that tumor and killing? Is this myeloid cell over here priming that T cell, or is it turning it off? What are the rules? What cells of there should we go after? And we're within reach of being able to do that now. That's what we need to be doing, and not just jumping and doing, all these giant trials. There are well over 3,000 trials now going on that I think are a waste of time. And they're big trials, and we know that 90% of them are going to fail. But if you go and you do 10 trials of 10 patients, you can avoid doing the ones that don't have a chance.
Thank you, Jim, for sitting down with me. This is a wonderful conversation and a fabulous reflection on your scientific journey. I really appreciate it. Thank you.
Thank you for joining us for this month's edition of All Things Cancer. Please subscribe so you don't miss future episodes. Learn more about how NFCR is working every day with the cancer research community to make cures possible at nfcr.org. You can also reach us on our call line at 800-321-CURE.